Hello everyone, and welcome to this overview of labeling strategies for your FIDA experiments. In this video, we'll be going through the parameters for selecting labels, as well as some of the multiple different labeling strategies available to fit your experimental setup. A FIDA assay is an assay based on fluorescent detection. As such, the excitation and emission wavelengths must be considered. There are 3 different available detectors for your FIDA instruments, and it is a simple 5-minute procedure to switch between them. The lowest available excitation wavelength is 275 nanometers. This wavelength allows for excitation of intrinsic tryptophans in your protein, and thus allows for label-free FIDA assays. The emission filter cuts off any light at wavelengths lower than 300 nanometers. The most common detector for the FIDA-1 is the 480 nanometer excitation source. This particular excitation wavelength is suitable for molecules that absorb blue light and emit at wavelengths greater than 515 nanometer, meaning green, yellow, or red light. This particular wavelength is suitable for most biological fluorescent tags and dyes. A 640 nanometer excitation source detector is also available, which sits in the red light range. The advantage of this detector is high sensitivity to red light fluorescent dyes, which has the lowest possible background in your buffers or samples. This is your one-stop overview of labeling strategies. We'll be going through all of them in this video. 3 general strategies can be applied when designing FIDA assays. You can either express your protein of interest as a fusion construct with fluorescent proteins. You can use chemical reactions to covalently label your purified protein. And lastly, a series of labeled interaction partner can be used as a form of non-covalent labeling. When considering assays performed directly in lysates, fluorescent fusion proteins are especially useful. These are directly expressed fused to your protein of interest, either in the N or the C terminus, depending on the expression vector used. As these proteins are born with the label, purification is not necessary, and the assay can be performed directly in cell lysate. A wide range of excitation wavelengths suitable for your detector setup is available. The most common fusions are green or yellow fluorescent proteins with the 480 nanometer excitation detector. The main drawback to this strategy is that the fluorescent proteins are 28 kilodalton tags and will thus represent a significant size increase to your protein of interest. A middle ground between chemical labeling and the fusion construct approach is using the SNAP tags. Like with the fluorescent proteins, you can express your protein of interest with a tag called a SNAP tag. It is a 19 kilodalton tag and is thus smaller than the fluorescent protein tags. When SNAP tags are applied, a large library of chemical fluorescent dyes are available, and the exact position of the label is known after using a chemical reaction to conjugate the dye. Moving on to covalent labeling. FIDA Bio sells 2 different labeling kits, both of which make conjugates with primary amines, meaning lysines and the N terminus. Therefore, it is applicable to all proteins. For this procedure, your protein needs to be purified and cannot be in a buffer containing primary amines. One of the 2 kits has a dye named ALC 480. The advantage of using this particular dye is that it is very insensitive to pH, temperature, and the local environment changes, meaning that any changes in the total fluorescent signal is a sign of loss of the labeled molecule, most commonly through aggregation. After labeling of the pure protein, it can of course be added directly to complex biological matrices like lysates. The second kit has a dye called FL480. It utilizes the same labeling chemistry as the ALC 480 kit and thus comes with the same advantages and drawbacks. The main difference is that the FL480 is very sensitive to pH, temperature, and the local environment changes, meaning that the binding related intensity change is likely to occur. So additional binding verification is available with this dye, meaning it is applicable to small molecule binding. If primary amine labeling is not suitable to your protein of interest, then thiol labeling is perfectly valid. The advantage of this is that you have much more control over where the label goes. The main drawback is that you can't use your reducing agents in your assays, and your protein must have at least one free cysteine. If you want complete control over where your label goes, as well as a wide library of commercially available dyes, then click chemistry is an option. This works by incorporating an unnatural amino acid containing an alkyne into your protein of interest. Thus, it requires special expression organisms as well as especially designed DNA sequences. Same advantages and disadvantages apply here as with all other covalent labeling strategies. If you want to investigate the interaction with peptides or nucleotides, these can also be labeled with suitable dyes. However, here we strongly recommend that the labeling is done as part of the synthesis when ordering the peptides or polynucleotides. Most synthesis companies offer this service for a fee. A single order of a fluorescently labeled peptide or polynucleotide will last for hundreds of experiments, as the FIDA only consumes 40 nanoliters of indicator per measurement, and the sensitivity is in the 10 nanomolar range. Moving on to non-covalent labeling strategy. The most commonly applied one is a fusion molecule of Oregon green and the tris-NTA nickel complex, which has some specificity for the his tags. Here the probe is used as an indicator and the target protein is added at a concentration where the tris-NTA is fully bound. This has the advantage that your protein of interest will be completely native, but adds an additional affinity constant to your assay design that needs to be considered. A quite important drawback of this approach is that the KD for the his tags is about 15 nanomolar, meaning that high affinity interactions cannot be characterized in this manner. Also, the tris-NTA has a range of unspecific binding to cell lysate proteins, and is therefore not suitable for this approach, and your protein needs to be relatively pure. If you need to characterize higher affinity interactions with a non-covalent label, then using a labeled nanobody or antibody is the way to go. The higher specificity of the antibodies allows for detection of low quantities of your protein of interest in buffers or complex media. The main drawback is that your non-covalent probe is now a relatively large protein, making smaller size changes more challenging to detect. Finally, we have the biomolecule-specific dyes. Examples of these include thioflavin T, specific for amyloid fibrils, and cybergold, which is fluorescent when it binds to polynucleotides. These two have previously been used for FIDA assays, but a whole range of molecule-specific dyes are available that are compatible with the excitation and emission wavelengths of the FIDA. The advantage of studying biomolecules this way, using FIDA, is that FIDA has no size bias and will give a more accurate measurement of a real molecule size. The main drawback is that most of the molecular dyes have a relatively low affinity and will need to be used at high concentration. This is your one-stop overview of labeling strategies. We'll be going through all of them in this video. 3 general strategies can be applied when designing FIDA assays. You can either express your protein of interest as a fusion construct with fluorescent proteins. You can use chemical reactions to covalently label your purified protein. And lastly, a series of labeled interaction partner can be used as a form of non-covalent labeling.